Hey, everybody. We're getting ready for, in my opinion, the highlight of the conference. Our own Dr. Tudge is going to share with us some things about hermit crab morphology. He's going to share some wonderful images that his students took under an electron microscope of specimens that were donated by our community to him. So this is really super interesting stuff. I'm going to read you just a little bit of his official bio because it's impressive. Dr. Tudge is a reproductive biologist with particular interest in the reproductive biology of invertebrates. His research focuses on the reproductive cells and associated structures, evolutionary mechanisms, and reproductive behaviors of marine decapod crustaceans. He also has experience dealing with other invertebrate and vertebrate groups, and his knowledge of reproduction in crustaceans can be directly applied to other taxa. He uses this interest in crustacean reproduction to invigorate. Oh, definitely. It was a Freudian slip, what can I say? The evolutionary history of particular crabs in the marine environment, and his work has direct application to aquaculture of commercial species and conservation of marine and freshwater crustaceans. He's also a keen local natural historian and bird biologist and teaches classes on ornithology, marine biology, and marine mammals. And we are just so honored and excited to have him. As you know, we met through the show that was broadcast earlier, and I'm so grateful for that, for having him join us. He respects the citizen scientist more than maybe anybody I've ever met, and hallelujah for that.
I don't have a whole lot to add, except for, to me, Dr. Tudge is like the Taylor Swift of the hermit crab world. And I am so excited that you're here. So I hope you guys all enjoy him. I'm going to be riveted. Please don't talk to me.
Oh, dear. Is this thing fitted okay? Can you all hear me? Yeah, is that pretty good? I'm glad you all got tickets because you can't get tickets to Taylor Swift. And can we dim the lights as much as possible to get these things looking a little better? I talk for a living, so this is really easy for me. I lecture at university in 2 rooms, 2 and 3 times the size of this, and I don't normally get mic'd up, but this will help with the electronic part of it here. It's a real privilege to be here with you guys. And as Mary said, we met through the work that Rachel Cusick was doing with that radio lab. And so it was fun to get connected. And I've been to 2 CrabCons before just virtually. So it's a lot of fun to be here in person for this first one here and to meet some of the names that I've seen and faces I've seen on screen at previous CrabCon events. And so I wanted to share a little bit of this with you because as I've done in the past, talking about workshops I've run at CrabCon about how you guys can collect information that's really useful. I mean, you know these crabs better than anyone else in the community, including scientists. And Stacey, I just wanted to say we're all citizen scientists underneath, even if we got robes or not got robes on. You know, everyone's a citizen scientist at heart, and that's what we should be teaching all of our children. And you should all respect the knowledge that you have from your animals, because you know them better than most scientists do. So it's nice to be able to use that information to collaborate a little bit. And I also thought this would be a fun thing to show you today. I am a microscopist by training. I did my PhD in microscopy. I trained on electron microscopes. They're sort of like, a couple of $100,000 worth of lenses and buttons to look at structures at incredible magnifications that you wouldn't get to see them under a normal microscope. And so, you guys might be looking at some of your larvae in your tanks, and some of you with really good eyes and some glasses might get a better look than other people. But I wanted to show you really what they look like at the microscopic scale and tell you a little bit about them, and maybe you'll understand a little bit more about what the larvae are doing and how important they are to the life cycle of your adult crabs that most of you have. So, all right, we got that up there, which is good. I also want to give a plug to the Crustacean Society. That's a group that I was president of a few years ago. It's a group of about 500 to 600 international crustacean biologists around the world, and I was just 5 weeks ago at a Crustacean Society meeting in Wellington, New Zealand. And it's kind of like this: a whole bunch of people get together, talk about crustaceans all the time, and everybody loves it. So it's a very similar meeting in many respects. All right, we'll start off at the very beginning, because this is important to understanding life cycles. You all know that arthropods, insects being a good example, don't grow continuously. They grow incrementally and have to shed or molt to get from one stage to the next. And crabs are arthropods as well. And so, and in fact, what an interesting thing we've worked out in the last 10 years, insects are basically flying crustaceans. They are each other's closest genetic relatives in many respects, which is kind of cool. They have really interesting similarities in their nervous systems. They both have antennae on their heads. Insects only have one pair. All crustaceans have two pairs. And there's other aspects of them that link the two together. But this molting process is important because you have to understand, you have to, during the life cycle, you're going to, an insect and a crustacean have the opportunity to change their form every molt. And we're so used to giving birth to little babies that look like little tiny humans with big heads, and they just sort of get bigger as they get older and hairier and fatter, but they look like babies, and your head stays the same size. But it's different for a crustacean, because a crustacean, hatches out of an egg. It may look nothing like what it's going to be as an adult and passes through these stages. And every molt, it's an opportunity to change its morphology and its form. And sometimes it can be incredibly drastic changes, and sometimes it's very subtle changes, depending on how many molts there are and what it has to look like ultimately. So here's a typical brachyurine crab and a typical shrimp with a couple of life cycles on there, just to give you some idea of what we're going through here, and to give you some idea of some of the pictures I'm going to show you about here. Of course, everything comes from an egg, whether that egg is internalized and it hatches inside a crustacean and comes out as a young, as a larval stage, or whether that egg is in the environment and floating around, which is what it normally is for most crustaceans. So the egg floats around and then it hatches, and the first stage larva is usually called, well, actually the first thing that hatches out, or not, but it's in the egg, is something called a nauplius, and that is something that unifies all the crustaceans. No matter what type of crustacean you are, whether you are a terrestrial woodlouse, whether you are a edible brachyurine crab, whether you are a microscopic parasitic copepod that's hanging on the outside of a great white shark and doesn't look like a crustacean, okay? They all pass through a stage called the nauplius. It's the first thing that hatches out of the egg, or the first stage that's in the egg. You might think to yourself, oh, you haven't seen a nauplius in my tanks. It's because the nauplius is in the egg and it hatches as a zoea, but the nauplius is there. So all crustaceans are linked by the fact that their first stage is a nauplius. And in fact, there's some famous work done on some parasites that live in the mouths of vertebrates. You might have heard something called a tongueworm. Okay, that's going to be found in foxes. It's famous in snakes and stuff like that. This thing is a horrible little worm, lives in the mouth or the esophagus or somewhere of vertebrates. And it was always assumed to be a worm. And when they hatched it out of an egg, a nauplius came out. It's actually a parasitic crustacean that lives in the mouth of a dog. Right? So think about that. But it hatched as a nauplius. That was the connection. Then they did the genetics of it and looked at reproductive biology and said, of course, it's a crustacean, but because it was such a modified parasite, you had no idea that's what it was. So the nauplius is a defining feature of all crustaceans. You won't see it in a land hermit crab because it's in the egg and passes through that phase to the first zoea, and that's what hatches out, the first little shrimp-like form there. And you have multiple stages of zoea, up to about 5. There can be more. These stages have different lengths to them. If you have less than the standard number, it's often referred to as abbreviated development, but it can vary depending on conditions and species and everything like that. And then you pass through that to a little stage that begins to look like a crab. Here it's called a megalopa. That's what's on your CrabCon t-shirts on the back, that little stage that's in the little fish tank there, right, little bowl. It can also be called a glaucothoe. The terminology, don't get too wrapped up in it. Everyone has different names for different stages, and you'll see here in some shrimp they call them a mysis stage and all sorts of things like that. But effectively, they're always egg, nauplius, zoea, megalops, and then you hit something called a juvenile. And we all know what juveniles are, right? They're under 18s, they don't get to drink, right? And things like that, right? So, and they're a big pain in the butt, right? So, but the definition of a juvenile is it has adult form, but it's not reproductive. That's the official definition of a juvenile. So when you go from megalops to a juvenile, it looks like a crab, but it probably doesn't have functional reproductive organs. So that's what makes it a juvenile. And then for humans, of course, we have to wait till puberty, till that turns on and everything like that, but these guys can be within days or weeks, can become reproductive. Not usually years, because most invertebrates don't live that long. But anyway, once it's reproductive, no matter what size it is, we call it an adult. And in fact, you could have a little tiny juvenile that can become an adult at the same size by simply developing reproductive organs inside it. And then you've got the definition of adult is after that, and then your adult can increase in size if you want. So this is a typical crustacean life cycle. And your land hermit crabs are no different in that. In fact, if we, what have I covered here? The only thing I didn't talk about, each of these stages is often referred to as an instar. So you'll go from a zoea to a megalops, and that megalops is an instar. Each zoeal stage is called an instar, and that's the terminology we use in a typical molt cycle, as it's called, for a crustacean. I've got to point it that way. Sometimes it bounces off the back wall, but you have to point it at the machine. Okay, so here's that same modified life cycle for your land hermit crabs. And don't forget, you guys are all into Cenobita and all the names you have for them, which I never get a handle on, Ecuadorians and purples and strawberries and whatever, that sort of thing. As a scientist, I'm just always, Clepiatus, Pilatus, all the scientific names in my head. But don't forget Burgess, the coconut crab, is the biggest land hermit crab. It is a shell-less hermit crab. And it's shell-less because it's got so big, it doesn't need a shell for protection. And remember, these all evolved on small, isolated, generally tropical islands with no land predators. And so they can get away with not having a shell. In fact, historically, before man turned up and introduced rats, pigs, dogs, mongooses, or whatever they put on, and humans, of course, to eat them, in some cases, the land hermit crab, the Burgess, coconut crab, was the biggest terrestrial predator of some of those islands. And I've seen them. They can be this big. They can weigh pounds, right? They can break broomsticks with their claws, right? Crunch, they're huge. They can be the largest thing. So essentially, they are a really large, shell-less land hermit crab. And their sexual biology is very similar. They just don't hang out in a shell, essentially. So this is a little closer to what I'm going to show you today. So, the females run down to the water. As you know, they release their eggs into the water. They immediately hatch into the first stage, zoea, and it's going to pass through 1, 2, 3, 4, maybe 5. It varies according to conditions and species and stuff. That's all aquatic. The glaucothoe, or megalopa, in this case, same name, same stage, different name. That's also aquatic. And then it's going to do its final molt into the juvenile, which is going to look like an adult, and it then becomes an asymmetrical hermit crab that then can fit into a microgastropod shell. And they find those tiny little microgastropods, because remember, snails have to grow up too, so they hatched out of an egg, and they were a little larva, and they developed into a little tiny form, and they start secreting their shell. So when they are microscopic, they secrete a microscopic shell. So if you've got a healthy population of snails living in the ocean, you'll have a healthy population of little tiny micro snail shells for these guys to move into. So they move into their first shell, and then they can live for a certain amount of time aquatically, but essentially what they're going to do is then move out of the water, because they now are, once they have an adult form or juvenile form, they actually stop breathing with gills, which is an aquatic way to get oxygen, and actually develop something called a pseudo-lung. They have a vascularized wet tissue, just like your lung, which you need to do gas exchange, and they can actually drown if you keep them in too much water, as many of you probably know. So they're eventually going to sort of say, I need to breathe more oxygen as they get bigger, and they have to leave the water to move onto land to take up that existence. And then everything's about water conservation, and breathing's not a problem because air has many, many times more oxygen in it than the same volume of water does. But it's all about, you're always losing water when you're gas exchanging across wet membranes. We do it. Every breath we exhale has moisture in it. You only have to breathe against a mirror to know that. And so we're constantly losing moisture through breathing. And these little guys do the same thing. So then they live in their shell, but they have to live in high humidity in places where water is available, either salt or fresh, because while they're breathing with their pseudo lung, right, they're losing water all the time. And so then after that, when they become terrestrial, it's all about not desiccating. That's important. So they'll often be nocturnal to keep out of the heat. They'll bury into damp sand. And that's why they often live, of course, in warm tropical environments, because 3 o'clock every day, just like Florida, I think I heard up there, you get a storm at 3 o'clock every afternoon, and you get your water delivered to you, right? So, so that's sort of what, yeah, that's right, set your watch by. And so that's when they take it over. And this one is, of course, the life cycle of Burgess here, the coconut crab. So eventually, that little tiny crab, right, dumps that snail shell as a coconut crab, and then actually becomes a little bit more symmetrical. You can, if you're brave enough to turn over a large coconut crab and look at it underneath, it actually looks symmetrical, but it actually is slightly asymmetrical in the way that it's built, and that's an indication of its hermit crab ancestry and relative to your land hermit crab. So this is the life cycle here, but otherwise it just, as you know, continues to grow, finds bigger homes, and works its way up with size and age through all of that. Now I have to give all the credit to where it's due. This is a wonderful undergrad, Anna Clark. She's coming on to be a third-year junior biology major. She's in our honors program. She's been working in my lab for a year now. She did all the work and took all the pictures, and I get to come here and have all the fun. But I did get her a bag to put some kitsch in it for her, so hopefully she'll be happy with that. But anyway, she got to learn how to drive one of these scanning electron microscopes. A little scary at first, because it's $400,000 worth of equipment, but it literally is a keyboard, a computer screen, and a mouse. And I thought, these children are born with that in their hands, so it was easy for her to do this. And so she also prepped all the specimens. Now, a scanning electron microscope, unlike a light microscope, doesn't use wavelengths of light to visualize something. It uses electrons, which get shot out of something called an electron gun. And essentially, you shoot electrons down a beam. You use magnets to focus it because you can't use lenses. It's all in a vacuum. And when those electrons hit your, I don't want to get too technical, when they hit your specimen, a computer looks at what gets bounced off and creates a three-dimensional image of it. But because you're using electrons instead of light waves, you can actually see things way, way smaller than you can normally do with your eye, because our eyes have evolved to see things using wavelengths of light or photons, which are bigger than electrons. And so we are limited how far down we can see, even with a standard microscope that you use light to visualize things with. Physics says we can't see it below the wavelength of light. But if you switch to electrons, you can actually see structures way, way smaller. And I'm talking tens of thousands of times smaller than you would normally see with your eye, and even an assisted lens through a normal microscope that you probably all used at school. Okay, so that's what an electron, in a nutshell, that's what an electron microscope does. Take away the light, use electrons in a vacuum, bounce it off a specimen you put, and then a computer's collecting all of the various subatomic particles that are being blown off this thing when you hit it with electrons, and it assembles a three-dimensional image for you to look at, which is kind of cool. You have to prep your specimens carefully, though. Unfortunately, they have to be dead because they can't be moving around. You have to fix them somehow. We normally use crude fixatives like alcohol or formaldehyde or something like that. But you fix it. You have to desiccate it because you can't have any water in there because it's going to be in a vacuum. All right? So you have to dry it out. And this is the secret part of it. If you dry things out normally, they will crinkle and crush and distort. So you have to use some pretty fancy physics and some fancy pieces of equipment, and you basically desiccate without crushing. It doesn't always work. I'll show you some pictures of some of these poor little larvae that have got a little deflated because of that. It doesn't always work, but essentially you are going to take all the moisture out of it, but you are going to retain its original three-dimensional form. Then you're going to shoot it with electrons, but you can't just shoot it with electrons. You have to have something that reflects the electrons, and we very coolly coat each of these specimens in a thin layer of gold, which is really cool. So each of these tiny little larvae is now sitting on a little metal stub, and it's coated in gold. It looks like a beautiful piece of jewelry posing in its position for you to look at. But that gold is there because you're going to hit it with electrons, and you're going to bounce some of those electrons off, and you're actually going to dislodge some of the gold, and some of those gold electrons are going to help you to make the picture. So that's kind of the physics of how that's going to work. So you have to fix them, desiccate them, coat them before you can even see them. And Anna did all of that hard work. It took her weeks to do that with these specimens you can barely see with the naked eye, and she had to mount them all with tiny tweezers and everything. And she turned out to be technically excellent at it, which is why she's still working in my lab a year later. And I've got plans for her with all sorts of projects coming up. And so, very talented young woman, and I give her all the credit for these wonderful images. And this is what you see. Now, it's black and white because it's electrons, right? You can false color them. You can get your program out, Photoshop, whatever, and you can make them look whatever color you want. But essentially, you're looking at the three-dimensional outside of one of these structures. These are little tiny zoea there. This is Clepiatus here. I think it's about zoea 3. It's a little tricky to tell stages from each other. I'll show you that in a moment here. It can be very subtle differences, but it doesn't really matter what stage it is. This is the zoea. They're all shrimp-like at this form, which is really quite interesting, considering what they're going to look like as an adult, right? These are one's laid out, one's curled up. You know, when you typically go and buy your shrimp when they've got been boiled with a little something tasty on the outside of it, like Old Bay, they usually curl up in that shape because that's how their muscles bend them in. This guy's looking exactly the same way, all bent with his tail over his head, hiding himself there. And here's one fully laid out, and I'll talk a little bit about their anatomy in a moment here. You're going to see a lot of images like this. This is the picture the computer takes, okay? A lot of technical information about how many kilovolts of electricity was going through and stuff like that. The important thing is magnification here. So this is 25 times its natural size, which means if you take that length from there to there and you divide it into 25 subunits, that's the actual size. Of course, remember, this is being magnified up onto a screen here, but this is 25 times its actual size. And then we also always have a scale bar here, because then if I'm going to take this away like I'm doing now and project it or print it at a different size, you can then use the scale bar to also calculate how big it is. This is telling you that length there is 500 micrometers. 1,000 micrometers is a millimeter. You guys never grew up with millimeters. I did, coming from the southern hemisphere. You got millimeter people as well? Good. Yeah, there we go. Millimeter. So we're looking at about, it's a little smaller than a 16th of an inch or so. So it's tiny. This is half of that. In reality, a micrometer is a millionth of a meter, which I love figures like that. They mean nothing to most people. It's like, you know, the US national debt. It's such a number that no one can really understand what it is. Right? Does it fit? If you stacked it up, is it on the stage here? Is it 2 stages? Is it 10 rooms? Is it 2 football fields? You know, it just gets so large. So a millionth of a meter. You imagine a meter from the floor to my hip, divide it in a million segments. That's the scale bar there. So you'll see the scale bar vary in size. You'll see the magnification vary in size, and it'll give you some idea of what you're looking at when you're looking at these sorts of specimens here. Now, we know about the phases, the instars that a typical land hermit crab goes to, because some of the pioneering crustacean biologists of the past actually went to the trouble of looking at them, describing, photographing, and drawing them, mostly drawing them back then, to give us these various stages. And even though Mary, mother of all hermit crabs here, is really the first to get them through to walking up onto land here, we mustn't forget that people like, I forget his first name. Anyway, Anthony, thank you. Anthony Provenzano also grew them up, hatched them, raised them, but all he was interested is in each instar, he took a couple of specimens out and dissected them and draw them, whatever. And then when he got to the megalopa or the juvenile stage, it was like they all got put in alcohol and got kept. And so we're interested in documenting the stages, and lots of scientists did this, and we have about about 8 to 10 Cenobita species, of which we know the full the larval sequence and what they look like at each stage, and we can all see how similar they are to each other. And there's a lot of crustacean larval biologists that do this sort of work. So we have to credit them with giving us something to be able to compare our specimens to. And you can see there are subtle differences between the various types of zoea. We're going to be looking at things like how many CD and spines are on their tail, how many sections are in the tail fan here, how big the rostrum is compared and the antennae and stuff like that. And you can see them all. This is the side view and the top view here. So we're going to be looking at a few of these pictures here, and it's not really important what stage they're at, but you'll recognize some of these differences as they go through from one instar to the next. You'll be able to see those sorts of differences there. So here's a beautiful single zoea all laid out here for us to look at, and it looks exactly like one of Provenzano's drawings there. So you see here, this one's times 40, so if you divide that into 40 segments, that is the original size of it, on the original negative that was there. So remind you about some morphology here. Typical crustacean, you have here something here called the cephalothorax, which is the head and the thorax combined. So this part and this part all squashed together. Okay, and then you have an abdomen at the back here, which is basically the tail in many respects, and right at the very end is something called the tail fan, which has a central piece called a telson and two little side bits, which are called the uropods, the inner and the outer, and that's the swimming structure. It's like putting on a pair of fins and doing the dolphin kick. That's what that is used for there. You've got a series of limbs under here, which at this point are feeding and swimming limbs. They are not walking limbs, but they are going to become the 5 legs of a typical hermit crab, of which 2 of them are used for walking, 2 of them are used for holding the shell, and then the largest set, of course, become the chelae, also used for walking, but we don't tend to call them walking limbs. We tend to call them chelae, chelopods, clippers, snippers, whatever you want to call them, all sorts of names like that. And then here's an eye here. The guy from London here was telling us about this. This one does not have an ocular peduncle yet. We're going to see that. But it is a large globular eye. I'll show you some wonderful pictures of that eye close up in a little while. It has this structure here, this huge rostrum, which if you think about it, you don't see that on your adult crab. They have a very tiny little projection between their eyes. It's, you know, but as a zoea here, they have this huge spike out the front of them, and it's actually mobile. It's hinged, so it can actually be raised up and down, which is quite interesting. And then underneath here are your 2 pairs of what you would call antennae. I would call the inner set antennules and the outer set antennae. They can sometimes be called antenna 1 and antenna 2, all sorts of different terminology, but you basically have an inner set, and that's the pair that you know that your crabs are doing this all the time with, right? It's like directing aircraft at the, right? They're sniffing, right? They're sniffing. They have a wonderful sensory organs at the end of that, and they're literally tasting the air, clearing it, tasting again, clearing it. All right? They're washing away the molecules. Otherwise, if you've got it's like having something up your nose and it completely blocks your nose and you can't smell anything else. So you have to get it all out, right? That's what these crabs are doing with their antennules. So they're sniffing, 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 clearing it, sniff, 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 sniff. And that's why you see them doing that. And the outer pair also is sensory, but they're more touch sensory than they are doing anything else. And that's a little different to their aquatic relatives who are doing a lot of tasting of the water column all the time and feeling pressure waves and things like that. But they can also use that to feel changes in the vibrations in the ground and stuff like that. So there's those 2 antennae, an inner set and an outer set, looking remarkably like each other at this stage, but you know they're going to look very different by the time they get to the adult stage there. So there's a typical zoea all laid out for you. Oh, I forgot to mention, also note this. You've got a spine here, 1 there, and 3 on this set. And sometimes there's a little one down here as well, and I'll talk about those spines in a little while. They look quite formidable on this little tiny zoea that's here. Here's a good example of that rostrum between the eyes there, right? But also note this. This is one that didn't survive the desiccation process too well. So this carapace has all been shrunken down during that desiccation process. Not all of them survived that. And so this has a little bit of damage from that. The eyes look fabulous, though. 2 large globular eyes, not on peduncles, not stalked yet, right? But still with remarkable, probably more than 200-degree vision from the front to the back. on this. And you can't really see it here. I'll show you a better photo later. This is the long rostrum here, and it hinges at this point here, so it can actually be raised and lowered on the head of that little tiny zoea. Well, I'll tell you. It's not a smeller. It's a defense. I'll get to that in a minute. So here's what that eye looks like. Remember the guy was telling you about all those little lenses that are in a typical eye? So this is an arthropod eye. It's a typical, what's called apposition eye. Each of these cylinders is a lens with light-detecting chemicals at the bottom of it. So each one is an eye. So each of those is the equivalent of your eye. So that is covered in hundreds of these, what are called ommatidia. And so anything that moves across the vision of this little zoea gets detected. So if something moved from downstage, upstage, it would be detected by this eye first, then that eye, then that eye, then that one, then that one. These eyes are designed to detect movement. All right? Our eyes are designed to pick up color and detail, and we pick up peripheral movement over here, but we're not very good at it. But your typical insect eye, your typical arthropod eye is there to detect movement. That's why it's so hard to squash a fly. No matter how fast you think you are, as soon as you start doing that, one of those lenses is going, something's moving here, it's coming closer, and then they're gone, right? So no matter how fast you think you are, it's hard to do that. So this is a wonderful eye for detecting movement. And at this stage, when they're little guys floating around the ocean, they're not interested in picking up really good images of stuff. They want to know who's going to eat them, right? Where they're coming from, and where they have to move to get out the way. And so this eye is designed for that purpose. And then the adult eye is built the same way, but it gets put on a stalk so that they have the ability to look 360 degrees with each eye stalk in a different direction. And it's the same sort of thing. They're looking for predators and things more than they are looking for anything else. So that's a typical eye there. Right, let's talk about these spines that are there. So here's another zoea here, and this is in the second abdominal segment. You can see it's got these three very distinct spines here. So these guys are really well armed when you think about it. It's got that big pointy rostrum, right? It's got spines all the way down its abdomen, and there's even spines on the end of its tail fan. And that's because this guy is a microscopic little piece of protein floating around in the ocean, and everything wants to eat it. Absolutely, especially tiny fish and stuff like that. So if they can curl their body up and project spines out, then that helps protect them against something that's going to try and bite them and engulf them. Probably not useful all the time, but must be at least useful some of the time, otherwise they wouldn't be there. And so the body here is covered in spines. Just about every body segment has got some spine on it for protection. And of course, you'll notice that all disappears when they become a megaloperon adult, because they're going to be leaving that environment of being plankton, and they become a substrate living animal, and then they become a terrestrial animal, and then their whole defenses have to become something different when you're on land, because there's no fish chasing you and stuff like that. So that at this point, they are heavily armored little tiny shrimp forms. And so you've got spines like that. You've got spines like this big curved one right on their back. And they don't look like much when the shrimp is laid out, but as soon as it bends into that shape, all those spines come out. And they're basically laid flat, and as soon as they bend, they start to project up like this. So they can be swimming, and then they can curl up and push all those spines out, like a pufferfish with all those spines, right? You know, when they're all laid back, they're not doing much defensive, but as soon as he puffs himself up, right, they all stand up, and they then have a better defensive function when they're elevated like that on there. So this guy's got a big spine on his back, pairs of spines down every one of the abdomen segments, 3 on the last one, and even some on the tail as well. And that big rostrum that you can point upwards if you want to, and point downwards if you want to, you can use it. And some of the brachyuran larvae, the zoea, the carapace is extended into spines that are 5 times the length of the body. So there's this little tiny crab that's like being duct taped to a 30-foot spike. And he's swimming around like that. That's because there's fish coming up to eat him, and they go to eat him, and there's a piece of wood this big they have to get in their mouth as well. Right? Or if they come at the front, they're going to get spiked by it. Or if they come from the back, they get spiked by it. So some of them really go overboard in developing these defenses. And you only have to look at a dozen different species of crustacean larvae or zoea. Every single one of them's got some sort of spike defense. So you know that being eaten in the plankton is a really big thing for these little guys. And so by raising them in the absence of those predators, they don't need these spines. You're getting way more young through than would normally survive. Somebody asked that earlier. We usually say it's probably less than 1% of all the babies that get hatched are ever going to make it out of the zoea into the megalopa stage to become adults. Because plankton is the food source. It's the pizza of the ocean. Everybody eats it that can fit it in their mouth, and it drives the whole food chain that's there. And these guys are a very large portion of marine plankton. Those little legs, these are the legs that they're going to be walking on at some point. But these legs that are underneath these little tiny zoea are swimming legs first. So this is what they swim with. They don't swim with that big tail at the end too much. They can do some with it. They spread it out, stop themselves from sinking. But they're paddling these things, so they have all these little tiny setae on them, right, that are like feathers. Each of those setae look like a feather, so they can spread it out in the water. They can lean on it. They can paddle on it. They're also not only swimming structures, they're feeding structures. So these guys, while they're swimming through the ocean, are grabbing microscopic pieces of plankton themselves and putting it in their mouths while they're feeding there. And so all those limbs that are underneath the body are all covered in these fine feathered setae that are used for swimming and feeding simultaneously at this point. There we go. So this is kind of the last stage, C5 here, most likely. Point out a couple of things here. You can see here's that spine, there's a spine, there's a spine, there's 3 spines there, there's one there, there's one there, there's one on each edge of that there, and then you've, of course, got the big rostrum pointing out the front there. And so they're a shrimp that's covered in this armor, and especially when you curl them up. That tail looks interesting. Let's have a closer look at that there. So you can see this is the telson, which is the middle part of that 3-part tail. And then you've got a uropod here that has 2 parts to it, and it basically fans out. That's why it's called a tail fan, and it can be pulled back in again. And so if they are going to swim with it, just like if you had a webbed foot, you fan it out, you push, you fan it back in, you do the return stroke, you fan it out, you push, right? So it can be used like that. And so that's why those 2 uropods on the outside can flip in and out and in and out. But you can see you've got a big spike there, one there, and there'll be one on the other one as well. These are not spikes in the middle here. They're actually setae. Again, they are spike-like setae, so they would have a defensive function. But this one here is actually an extension of the carapace. It's as hard as the rest of the carapace is. So it's a true spine. The others are actually spine-like setae, and some of them even will probably articulate at the bottom, and they could probably mobile. That's one of the definitions of the difference between a seta and a spine. A spine is immobile. A seta is mobile of some sort. And like I said before, we saw feather-like setae. Setae can assume all sorts of forms. We've documented more than 200 different types of setae in crustaceans alone. So that mobile structure can be feather-like, it can be a sensory structure, it can be round, it has all these different structures and forms of that. So far we've been looking at zoea here, zoea 1, 2, 3, 4, and this one. And then finally we get to the megalopa, or the glaucothoe stage, which begins to look, at least the head, it now begins to look like a hermit crab. And so you've all been waiting to see this. Here's your first look at a little tiny crab, all right? Now, first thing you notice, look at his eyes. From C5 to megalopa, his eyes go from being globular to being pedunculate. They now have an ocular peduncle on the bottom of them. So this round bit here is where all the ommatidia are. This is the lenses, and this is just the stalk. So now they're now raised up on stalks, and each eye can be moved independently and put down, and they can look behind them and forward at the same time, and they're like 2 little telescopes with their lenses all on the end. And all the adults are like that as well. So this is the first time they get an adult-like eye from that stationary globular eye that they had in the zoea stage. You'll notice that the 2 antennae start to look different from each other now. Here's the inner pair, the antennules. These are the flicking, tasting ones. And here's the outer one here, which is going to be more the whip-like feeling one. So they've all, in one molt, you now differentiate those two antennae more than they were before. So the eyes are now different, the antennae are now different. And now look at these limbs. They're no longer little fluffy, feathered, covered feeding and swimming structures. You've now got these robust walking limbs with all sorts of setae on them and corneous little tips that they can stand on with spines on them. And, of course, the first set become the chelae or the claws for manipulating and feeding and all the things that they're going to do there. So, and you can see this guy's carapace is crushed a little bit too there. Sometimes Anna had to pick it up with a pair of tweezers to put on there. Sometimes that's tweezer damage too. So that could be one or the other there. But looking remarkably like the hermit crab and remarkably like what you're going to see poking out of one of your little shells when you stick your head in there. And this is just a side view of the same thing to show you, here's now the back two legs here, what do we call P4 and 5, right? P1, chelae, P for paraeopod, which is our term for one of those legs there. P2 is the first big walking leg. P3 is the second big walking leg. That's all the part that's outside the shell. And then you've got a little pair of almost like T-Rex arms, right? Right back here. P4 and P5 have been modified to grip inside the shell now. Okay, so they don't look like, here's P3 here with a dactyl on the end, a little claw there. These guys now have a modified end to it, even have little rough pads with scales on them that they use to grip the inside of the shell with. So they're using those last two pairs of legs for gripping inside the shell. That's what they pull themselves in with when they pull themselves back. All right, they're using those two sets of limbs to pop themselves in and out of that shell. So you now got also modification of all of the walking limbs and the chelae from the last Zoea stage as well. So this is a major change in morphology in this instar from one to the next. But this is still a symmetrical shrimp-like body though. Right? We still have discernible segments here, all the abdominal segments and the tail fan, and it looks like the back end of a shrimp or a lobster or a crayfish, and it's symmetrical at this point. I think I put in, for those of you who are interested, now I've got a few pictures of some Perlatus. That was all Clepiatus, so here's a couple of Perlatus here. We're going to go back. This is Zoea 1, very first stage. Look how simple the tail fan is. You don't have a telson, you don't have uropods yet, and you've only got little spines and a few setae there. That's actually its butt there, right? That's its little anus down the very end, last segment there. Stays there for its entire life, doesn't move anywhere else on the body. But you'll see how simple the tail is on a Zoea 1 to how different it was when we looked at the Zoea 5 with all of those uropods that flick out and all the spines and everything there. So like I said, every instar, there's some sort of morphological change going on, even though they're kind of shrimp-like, and not. You've got all these changes that are going on there. This is the best shot I had of that mobile rostrum I wanted to show you. This is a Perlatus Zoea 2, so quite early on. But you can see this hinge here. This is where this little rostrum actually hinges and is capable of moving there. You'll see this eye is a little sunken, so it didn't survive the desiccation process too much. And you can see how tiny the little antennae here are. So here's your antennules on the inside, and here's the antenna on the outside, and they're quite similar to each other and quite small, and they're going to change drastically as well. So here's your megalopa of Perlatus here, looking very similar. You can't really distinguish them at this point, one species from the other. It comes down to counting hairs and setae on limbs like Provenzano and these people had to do to tell species apart. But you really can't look at something like this and say, oh yeah, that's a blah blah, right? They all look pretty much the same. So same sort of thing, we're looking at all of those limbs there hanging out. And this is just a better shot of how they have that segmented, symmetrical abdomen, just like shrimps, lobsters, crayfish, and stuff like that. But don't forget, you then have to, this is being recalcitrant. Remember, your shrimp-like Zoea becomes a lobster-like megalopa, right? But it's going to eventually become this juvenile that's asymmetrical, and the abdomen is going to lose its fundamental segmentation. Each of these body segments is left as this tiny little line there and there and there are the only vestiges of any carapace that's left, any shell that's left on them. So basically what happens is between every body segment is a flexible membrane, It's a bit like a suit of iron, not iron, a suit of armor. That's the word I was after. Thank you. A suit of armor where you can only bend at the articulations, right? So every body segment is hard, but the articulation is a flexible membrane. So what the abdomen does is it decreases the calcium that's in the segment and increases the flexible membrane in between, and you end up with a very flexible body with no calcium left on it, because it has to become asymmetrical to be able to twist into that typically dextrally coiled or right-handed coiled shell. Somebody this morning mentioned the sinistral, or the left-handed, and it was the church that called it sinistral because it was sinister, left-handed, right? There's only about, I think it's about 4% of all mollusk snail shells in the world are left-handed. All the rest are right-handed coil. So they're developed for that. You can see all your same structures you saw in the megalopa are all there. P1, P2, P3, P4, P5, little tiny T-Rex limbs at the back here. And then you've got this curve. And then you've got your tail fan. It has the same structure, but it's greatly reduced. There's a little telson, little tiny uropods. And you can actually, this is the only place on the abdomen you can still see asymmetry in a hermit crab. In here is, if you pull this abdomen out, it has some little structures on one side that are not on the outside, that are not on the inside. So because it curls it inwards, it reduces the structure on the inward side. And so things are smaller there than they are on the outside. And so you see that asymmetry there. And that's because they're going to be hauling themselves into that coiled shell and eventually spending their whole life in it. So this is a switch from symmetrical larval stages to the asymmetrical juvenile that's asexual to the asymmetrical adult that is now sexually active, and so therefore is an adult now. So that's the process of getting their bodies there. Now, I have to thank Anna Clark. She did all the work, right? Andrea Brothers is my very talented microscope technician who also trained Anna to be able to do all that stuff, and she keeps all the microscopes up and allows us to use them whenever we want to, etc. So I wanted to thank her. And both Mary and Stacey provided the larvae that you saw there, sent in the mail, and so I hope I put them to good use. And I'm going to continue to do some of this work. I've got more undergrads coming through that I want to put some projects onto and do some microscopy and stuff. And I have happy to take some questions here, or else have a few more slides if you want to see some of different structures on adult hermit crabs, if you wanted to look at those as well. But I thought while you've got Zoea in your head, I want to take 5 minutes to talk about questions about the Zoea pictures I showed you. Anything that you have about, any questions you had about that? Any of those, the process or any of those structures or anything like that? Yes?
People say a lot of times that hermit crabs don't have good vision, and that's just not my experience, and especially what you showed up there. To me, that says specific vision, but highly developed. Would you agree with that?
Absolutely. I think there's no such thing as poor vision. It's just different vision, right? We have wonderful 3-color vision. Like dogs see well. They don't have 3-color. Sharks don't have 3-color. Everybody sees well. Evolution gives you an eye that's useful, right? They have a perfectly useful eye. Now, they don't look at a Mona Lisa and can see all the details that are there and everything, but if that Mona Lisa moves a millimeter, they know it, right? So their eye is incredibly functional for what they need it for. It is a highly evolved, the arthropod eye is a highly evolved eye for both acuity and movement, but it doesn't have the same abilities that our 2 lenses have for us. We use them differently. So I would say there's no such thing as poor vision. There's no such thing as an underdeveloped eye or a bad eye. They're all good for whoever's got them. And it would only be mutation or bad development that gave you a non-functional eye. It's all there for a good reason. Yeah, you want to send the mic around? Yeah.
Okay. I'm just trying to understand all this.
Yes.
You had said that when, like you said that they hatch immediately. So when the spawn occurs, do they pretty much hit the salt water and hatch? Which would imply
I think
that
that's
they're
true.
in that
I don't have any
nucleus.
experience raising those larvae, but there's no reason for them to be inside there because they have very little yolk They're already gone through the nauplius stage. They need to be out and feeding as quickly as possible. So they can't sit inside that egg for any length of time. They have limited oxygen supply. They have limited food supply. So by immediately, we're probably talking minutes that any egg would last before it hatches, because otherwise the little zoe is going to run out of resources, right? You're trapped in a small room with nothing to eat or breathe. You've got to get out as quickly as possible. And so then they become a feeding stage. Yeah. I'll put something prettier for you to look at for a minute. All right.
I was just wondering, why aren't there any stages between the fourth zoea stage and the first megalopa? That just seemed like a really big jump.
Yeah, it's amazing, isn't it?
Okay, thank you.
No. Well, it is. You're looking for incremental change. Well, sometimes you don't get it, and that's a major morphological change. There are examples of even greater morphological changes in the animal kingdom and in invertebrates, where you go from something that doesn't even look like what it's supposed to what it's supposed to in a single change. So some invertebrates have the ability to literally reprogram all their cells backwards to become a mass of primordial cells and grow again from that without having to go through any intermediate stages. So that's a big jump, admittedly, but in the scheme of things, it's not. So I didn't mean to belittle that, but no, that is an amazingly large change in difference. Now, some have multiple zoeal stages, more than 5, and sometimes they extend them out, and the length of time their zoeas can vary, etc. It all depends on resources and time in the environment and their genes, etc. So you might end up going through way more zoeal stages, and you might get incrementally a little closer to that, but there's always a big jump to megalopa and glaucothoe, and then ultimately onto juveniles and adults. It is quite interesting. It's just the way it evolved.
I will add, too, that is a stage where we lose a lot of them. It's such a hard transition, just getting out of that old skin into all those legs and everything. We lose a lot at that stage.
Yeah.
I actually just had more of a technical question. You were referring to a lot of the instars as like C4, C5. What does the C stand for?
Sorry, the C was the Annis code for Clypeatus,
Gotcha.
and P was for Pilatus. So she took all the pictures and coded them all on the negs, which I told her to do so that we could look at any negative and know what it was, even out of context with the rest of them. So she had a little system of naming them, and it was C for Clypeatus, P for Pilatus, and she didn't use Z, but she used 2, 3, 4, 5 for the zoeal stages, and then M for the megalopa. So it was a code that she had. Yeah.
Thank you.
Is that my time? I got to leave?
No.
So you talked quite a bit about vision and sight, but what about hearing structures? Do they have any type? I've noticed mine don't seem to respond to sound. So I'm wondering, is there, like if you move an inch, they run and scurry, but like if there's a loud sound or something, they just don't seem to react.
If the sound is not transmitted to the substrate, they won't feel it. Hearing is simply deforming molecules of the air that then move hairs in our ear. They have hairs in their body that are deformed by the substrate. So I'm pretty sure they can hear in the air reasonably well. You have to produce a large deformation to have it shake things on their body from there, but they're more likely hearing through the substrate. And I know that they chirp, and I know that it's been shown that they can communicate with one another through the substrate. Many crabs can do that. Fiddler crabs do the same thing. They tap on the substrate, and it gets transmitted through the substrate. So that's hearing as our hearing. We think of aerial hearing. It's deformation, sound waves in air. You put sound waves through something else, including water, sand, soil, rock. That's hearing too. So they're perfectly capable of feeling all those sound waves. They may not react too much to sound. Probably they're quite muffled inside their crab-habitats and stuff like that. And I'm sure that you guys are not deliberately producing large sounds. You don't want to scare them. They can probably hear you fine.
Across the room on the TV or something that we're like, and there's no response.
Yeah. No, I think if you were stomping around the house or something like that, they would feel all of that. So they.
I have my surround sound speaker beside me.
They're probably rocking out on that. Yeah.
Do they actually have a brain, or is it just ganglion?
There's really no difference between a brain and a ganglion. A brain is a concentration of ganglia that is mounted inside the head. So you can have a distributed brain. Computer people know that. You can have a distributed brain. So that can be multiple ganglia down the body. Each ganglia is functioning like a little brain. But in the evolution of animals, one of the first things they did was something called cephalization. You're going to make a head. The part that meets the world first, you want to concentrate all your sensory organs in. So that's when the sensory system was all concentrated in the head, produces a head. Cephalization and a brain is simply the cephalization of distributed ganglia. So yes, they have a brain. They also have they have a brain halfway down their body and another little brain in their abdomen. And so those little brains can process stimulus from nerves themselves and send back without going all the way to the head. We tend to have bodies that don't do that. We tend to process everything in our head. That's where we concentrated it. But literally, they have multiple, kind of by definition, multiple brains in their body.
That's so interesting.
Brainy little guys.
How do they make like the chirping sound?
I believe there's multiple ways they do it. I believe they have a stridulating apparatus, which they rub, and I think that's on their cheela. Those of you who have seen that, I think it's on their cheela. They have a rubbing apparatus there. They can also probably rub any of their limbs against any other limb to do that. And there's usually, to make the best controlled chirping sounds, you need a structure to rub against another structure, and I believe they have these ridges that they literally rub limbs together. But no, they're not. It's not sound produced anywhere within the body. It's rubbing.
Anybody else? Now's your chance.
Do you want to talk about adults first before we ask questions about adults?
Well, I was just going to show you this little thing here because this is kind of fun. With some more, it's got some more electron microscopy in it, which is kind of cool. Oh, one more question back there. Okay.
I actually have two. When you were talking about the antenna, I've noticed my crabs will put their eyes down and wipe the antenna over it. Are they cleaning their eye?
Yeah, that's what most of them do. They will clean all of their sensory structures on a regular basis. They'll pull their antennae down and rub it through their mouth, and they can't get their eyes in their mouth, so that then they wipe it with their antennae. So it's what they've got to clean themselves with, essentially.
Okay, and the second question I had, is it the last set of legs they use that so many people think it's alien looking, where they're cleaning out their shell?
I would presume so. I've never been in the shell of a hermit crab. So
you see something moving that's...
Yeah, it's most likely going to be those last two pairs of legs that'll be used for any of that sort of stuff. Unless you've seen them come out of their shell, stick their other legs in to hook stuff out, but normally they're using their body that's in there to get stuff out. Eggs that fall off or don't hatch, or remember they poop back in there, so they got to get that out too. Yeah. They got to clean house, empty the litter box, and that sort of stuff. So, so it is most likely that 4 and 5. They got no other structures back there to do it with except them, right? So I put a few extra slides here together because I also wanted to recognize some other work that I'm doing based on a member of your community. And if Mo is listening right now, I just want to say thank you for stimulating this research, Mo. This is Mo Lundberg from Sweden. And the first CrabCon I went to remotely, Mo and I talked a little bit afterwards because I said we were talking about something, and she said she knew her crabs really intimately. And she started talking to me about photographing structures on her crabs that she said were the same between molts, so she could track individual crabs. And I thought this was so cool because that's one of the problems we have with some of the scientific research, is knowing whether you've got the same crab, because they switch shells all the time, right? And how do you know, if you want to track one crab and know what it does, how do you know it's the same crab, sort of thing? So she was talking about that. And then spun off out of this, Moa sent me these pictures one day, and she said, I was looking at my crabs. She takes these beautiful portraits of her crabs close up, and she said, what are these two holes? What looks like a gland and a tube, and this hole on the antenna here. And she sent it to me with these rings around it, and I looked at it and I thought, oh, well, that's an antennal gland. Of course it is. And then I looked it up and I thought, no, that's the wrong limb. It's supposed to be on the other one. It's not supposed to be on the antennule. It's supposed to be on the antenna. And I thought, oh, I don't know, hang on. So I flipped it to a friend of mine in Georgia who's a crustacean physiologist and does all the gland stuff and everything. And I said, you ever seen this before? And he said, no, what's it on? And I said, it's on a land hermit crab. He said, I've never looked at land hermit crabs, but I've never seen anything like it on any crab, any lobster, any crayfish or anything. I thought, oh, that's really interesting. And so I said to Mara, look, I'll look into it a little bit. And she sent me pictures of all of her species that she has showing, and I had to reduce them down to get them all on there, but they all have the same structure. So this is multiple species that she has. She's taken all these wonderful portraits of them, and they all show the same structures in there that we just saw before. On the inside, like if this is the antennules, right, they flick them out like this, it's here, all right, whatever segment that is, it's the third one back. And then I went and looked at Burgess. I went to the Smithsonian Institution. I went and got their coconut crabs out of the collection. I pulled a couple out and I put them under the microscope. And sure enough, they have the same thing. There's one there, there's a gland, and I thought, well, that's really interesting. Burgess has got it, Cenobita's got it. I wonder if it's just in the land hermit crabs. So the first project Anna Clark did for me was an antennule structure project. And she produced a beautiful poster at the end of the semester, which I then stole some pictures from to show you a little bit about this this structure here. So she put one of these antennules of one of the, I only had 2 specimens of some land hermit crabs, really old ones from the 50s, in a jar in my collection. I have a huge collection of invertebrates because I teach with them. So I went crawling through my tubs of jars of specimens looking for, did I have an LHC in there somewhere? And I pulled it out, 2 of them in there. So she got to pull one of these off and she put it underneath the electron microscope here. And so this is this part here, and this is the part that goes up, and then the little bent part at the top, and then this is the cool little structure that they sniff with. All right? And I'll show you some more of that later. But she said, look, I think there's a hole there. This is right at the base there. But we had a look at it, and that was actually a rip. And we looked at the top part here, and there's no hole there. There's no gland there. This is all membrane. Remember how I told you between each segment there's a membrane so it can articulate? Somehow on these guys, that piece of membrane has been extended down and runs all the way up the full length, and I don't know why. But it's just a large membrane patch here. This is not calcified, it's flexible. Now remember, they're flicking these things constantly. So I'm trying to think of why they would have that, but I still don't have an answer to it. Next thing I want to do is I want to slice this way and look inside, because if it's a gland, you'll see it, right? And I might even try and slice it this way and see what it looks like. But I got to get fresh specimens for that, and as I've told the community before, I'm not going to go and buy any and terminate any for science, but if you guys have some that are freshly deceased and you want to donate them to this, I'm happy to receive them, and I can use them for my research. And Stacy has sent me some, no, not Stacy, Dan and Sam, sorry, Dan and Sam here, Samantha and Dan who are here, vendors somewhere? I think they're at the vendor. Oh, they're at the vendor there. Anyway, they recently sent me one of theirs that was recently deceased. And the thing is, they can't be too long deceased because otherwise the tissue rots. So I know that's sometimes difficult. You put them in alcohol, put them in the mail. I can send you instructions.
What if
Yeah.
you lost one over the winter and because you wanted to bury it, you put it in your freezer and maybe still have it?
The freezer? My freezer's full of dead birds, so I completely... I'm fine with that. No, the problem with freezing is it distorts the tissue when you thaw it again. Ice crystals form. Now, I definitely can't use frozen tissue for electron microscopy. It would give me a crude look at something, but it's not as freezing is not the best way to preserve that tissue. So basically, you go to the CVS. You can buy rubbing alcohol, which is anywhere. It's usually 90 to 98 or something % alcohol, and you can water it down a little bit to about 70%, and you basically soak the whole animal in it for a few days. You need about 5 to 7 times the volume of the crab of alcohol there, and then you can take it out of the liquid. You can wrap it in something like paper towel that's just got a little bit of alcohol in it to post it, because you're not supposed to be sending liquid alcohol through the USPS, right? But it can be preserved, and it can survive the few days damp until it gets to me, and I'll put it back into alcohol when it arrives to me. But if you want instructions, contact me. Stacy's got instructions as well how to do that, and I'd be happy to use any of your passed on pets for research if you're willing to do that, because I don't want to go out and collect any from the wild or get any from any pet shops or anything like that. So it's another opportunity for you guys to engage if you would like to do that. And so we looked at this, and at the moment it looks like it's not a special gland structure, but we also don't know what it is. And so I'm going to do some investigating of this and look and see what structures. Because if I can look inside at the cells that are there, I can tell what it is. That segment should be full of muscle. That should be all that's there, because it's being used to operate the rest of the antenna. It should be a big old muscle in there, maybe multiple muscles. And glands look completely different from muscle. Under the microscope, I'd be able to tell them apart. So it'd be kind of cool to have a look at it and see what it is. But at the moment, it doesn't look like it's a gland, but I don't know what it is. So that's kind of cool. And one thing I also asked Anna Clark to do is that I told her, let's look at aquatic hermit crabs and see if they have the same thing. So she went and looked at a diogenid in the family Diogenidae, an aquatic hermit crab, and a pagurid in the family Paguridae, both saltwater aquatic hermit crabs, and she did the comparison of the same limb between wet, dry, and wet, right? So this is just Cenobita down the middle here, and she looked at that segment and found that in both the two families, they did not have that structure. This seems to be a structure that's unique to the land hermit crabs, but I still don't know what it is. And I also want to show you here, this is the end of the antennule that's used for tasting water, and this is the same structure that's used for tasting air. And you'll notice that these have long, the sniffing cells are inside all these setae that are here. And aquatic hermit crabs do the same thing. They wave them around, clear them, wave them in the water. They're tasting all the time. But you can't have a mop like that. You all know when you take a mop out of the water and hold it up, all of the things flop down and you can't do anything with it. So a land hermit crab has completely modified that. So these hairs here are a tiny little brush at the top, and all these segments are these segments here. So they've gotten rid of all of the hairs because they don't need them to sniff air. Right? It's been modified for aerial sniffing, not aquatic sniffing, which is kind of cool. So you can see the same end modified for terrestrial existence, essentially. But this is something that Anna's been working on, and I'm going to continue working into that. And I think I didn't have any more. That should be it. Yeah, I think that's it. So if we've gone way over, but I'm happy to take any questions if you got any. Yeah.
I don't know how dumb this question is, but
no such thing as a dumb question.
Can hermit crabs heal the soft tissue versus having to molt it?
They should be able to.
Okay.
Most of the hard tissue gets healed at molt. Some of the soft tissue can probably be healed without molting. Yeah.
I have the talk open, or I have the chat open, and Moa was saying that,
cool.
The openings that were circled in the previous slide with her picture, Tammy White's talk, her and Tammy's talk from last year on anatomy, they found out that those openings were the openings to statocysts,
cool.
Which is the organ for maintaining balance and orientation, and which would be probably related to how hermit crabs, I remember there was questions on, how hermit crabs know which way to dig up. And when they're underground, how do they not get lost underground? And that's kind of how they maintain balance and orientation and stuff. And
cool.
Moa wanted me to,
cool. Thank you, Moa. Thank you. Well, that's good. I'm still going to look at it, though. I think it'll be cool. Because if I can slice through a statocyst, I'll actually be able to see it. And what it normally is a hollow ball that has hairs poking on the inside, and inside it is floating a little hard ball. So essentially, whenever it moves, the ball goes and tickles the hairs. Yeah, in a way. Mm-hmm. Although we have 3 different direction channels in our head that do the same thing with liquid. We have a bubble in there in the liquid. So your eustachian tubes, your circular channels, tell you which way your head is, and that bubble moves around in there. But this has a solid ball, usually in a fluid-filled sphere. And as the ball knocks from one side to the other, it stimulates the hairs. That's normally how a statocyst works. So it'll be cool to see that if it's in there. So that's cool to know. Yeah.
Real quick, so I know you were talking about in the beginning, they did have the gills to be able to breathe in the water. But where did those morph into this that they now turn into
wrong way.
lungs?
There's one picture here that has gills on it. Gills appear on Zoea 5. Come on. Where's Zoea 5 here? Come on. See that there? That's a gill. That's the start of a gill being produced. It's underneath the carapace, right? So it's each limb, each of those little tiny feeding limbs has a gill at the base of it. It's a little fan-shaped structure that does gas exchange, and it's protected by the carapace, but the water comes up underneath and circulates there, and they breathe with it. And then essentially that gill completely disappears in a megalopa. It's not there anymore. You can find a megalopa. This is not being very... I wonder if my battery's going. Could be. Megalopa. Maybe I'm just overloading it. Yes, I am. So here's all of those limbs here. There's no gills there anymore. They might just be there as tiny little bits, but they're not poking out anymore. And the lung develops in here. It's an expanded piece of tissue that's under the carapace. Gets vascularized with blood vessels, just like your lung, and it gets kept wet, and that's where the gas exchange goes on. So the gills reduce, and the lung starts forming probably at about the megalopa stage. It's switching from one to the other, and definitely by the time it's a juvenile, it's probably got a lung and not any gill vestiges at all, which is why they're like, got to get out of here, right? Got a shuttle filling. Oh, yeah. Oh, absolutely. Yep. Oh, cool. Yeah. Yeah, there was some work done years ago in Australia where a researcher was perfusing the lungs, so she was filling the blood vessels, the arteries and the veins with rubber, different colored rubber, and she was able to then show the arteries forming over one half of the lung and the veins over the other half, and so that's where the gas exchange was going. It was really cool research. 90s, I think, maybe early 2000s.
Do you need marine crab specimens or only land crabs?
I have a pretty good collection of marine crabs, so even though, oh yeah, why not? Sure. But remember, ideally they should be collected ethically, which means if wherever you're going, you should probably have permits for doing it and stuff like that. I mean, the average person shouldn't be going out and collecting 200 aquatic hermit crabs and bringing them home and posting them to people, right? So, just remember that. You guys all have very good reasons to have your crabs, and I just wanted to make use of any that were no longer of use to you. So, questions?
So you talked about the two sets of antennae and how one is for tasting and how one is for touching. I think all of us as crab owners have witnessed some pretty epic, we'll just say, antenna fencing. And nobody really seems to have a concrete answer as to what is happening. As a scientist, what is your best guess speculation on occurrence of that?
So firstly, probably both antennae do both, but the antennules are primarily sniffing and the antennae are primarily feeling, but they probably have sensory structures for sniffing on both and sensing on both. They just do different things with them. As the Crab Museum guy said, in aquatic decapods, they have a gland around their mouth that they basically pee out of. Their urine gland flows into the front of their mouth, and then they push water currents out with it. They squirt it out, and that's how they communicate with one another. It's literally a pissing match between two crabs, right? And they're sending chemical signals in the water. Well, they can't they can't do it with a liquid, but they could be producing chemicals, because they do detect aerosols. They can smell dead hermit crabs and go because they know there's a shell available. They can smell food and direct it. They've done all those sorts of experiments. Those wonderful Y experiments where you put a crab at the end and you send food down one half, and then you count how many times it goes to the food and how many times to make sure it's not random. And they do all sorts of experiments like that, and they know that they can orientate to things they smell in the air. Now, whether they're producing their own smells and therefore that's what's going on there, face to face. But they, I bet they're getting closer and doing a lot more sensory stuff. I've never seen two land hermit crabs duking it out. So are they literally touching each other and their antennae and antennules? Bah, bah, bah. So to me, that sounds more tactile than it is, because, I mean, ultimately, the last thing you want to do is get hurt, right, or damage yourself over any fight for territory, shells, or girls, right? You don't want to die for that. That's not selected for evolutionarily if you have to die. So that's why a lot of contests don't involve physical stuff. It's, I'm bigger than you, I smell worse than you, you know, all that sort of thing. I'm more colorful than you are, and it very rarely comes down to physical combat, because the last thing you want to do is lose a limb. So you can imagine, exactly. So I can imagine a couple of guys getting together and just going, back off, back off. There's all sorts of verbal stuff going on there, you know, and then they don't actually, it would only escalate to that as any sort of physical stuff as the last resort, most likely. Sounds like it's tactile mostly to me, but we don't know if they're producing anything out of, you know, they could literally have converted those glands into sending out stinky smells that they're directing at one another. But you would expect them to do that at a distance a little more than what they're doing physically touching. So I've never seen it. My guess would be that's tactile boxing and not necessarily chemical, but there could be a chemical component. Yeah.
I have a ton of videos of them on the wild. So they're hanging out here. There's like 2 of them are right there.
Yeah.
So that's
and I would assume that at mating time, the boys are doing it to one another. They might be doing it to potential mates. I can imagine that they try to intimidate somebody they want to take a shell off. And so it's probably for all of those behavioral functions. It's sort of all they got. They've shown in aquatic hermit crabs that size is really important, and the size of the claw is really important. And often the two crabs will just touch each other for a little while, and the smaller one will back off, because what they've done is they've assessed each other and they've been measuring your claw or your leg or whatever, and he knows the little guy knows the other guy is bigger than him and backs off. Exactly. So a lot of that is that assessment without conflict that you want, because that gets rewarded evolutionarily, because conflict won't get rewarded. If you die out of a conflict, then there's no message to be sent on in your genes to the next generation. So, there might be some of that going on as well. It's common in aquatic crabs anyway.
I think we might have time for one more, if anyone has one. But we could just listen to you. I know. We could. Yeah.
I want to know how you have all that information in that head of yours.
It's a sponge. Yeah, but you know what I can't remember? That I'm supposed to take my wife to the movie next week and stuff like that. So I have a head full of useless information, but I forget names easily, like people's names and stuff like that. I sectorize it like a computer does. You know, put it in different places and recall it out when I need to. Yeah, I've cataloged all there very carefully. And I can remember stuff that I learned 30 years ago, but like I said, I can't remember what I had for breakfast yesterday. So, well, anyway, thank you very much for the opportunity to speak today.